Okay, here we go. I'm so excited to welcome David Higgins. And he knows I'm so excited because David's awesome. He is at the Center for Data-Driven Discovery in Biomedicine, the Scientific Community Program Manager, and is here to talk to us about some really fantastic tools that are in practice and how to use them, whether you are the point and click type or you really want to get down and dirty with the data. So David, I will hand it on over to you, my friend.
Perfect. Thank you so much, Eden. So let me share my slides. So thank you so much for welcoming me to speak today. Can everybody see my screen all right? Eden, can I get a thumbs up or a yes? You can see my slides?
Looks great.
Looks great. Perfect. Okay, let me click that button and wait for it to go. There we go. And there we are. Hi. So thank you so much. So hi, everybody. So my name is David. And I'm here representing the D3B, the Center for Data-Driven Discovery in Biomedicine, which is based out of the Children's Hospital of Philadelphia. And so thank you so much for inviting me to this event. And so it's a great cause and a great purpose. And I love talking about our tools and ways that we help researchers and clinicians learn more about rare diseases that we reach as part of our platforms. So today I'm going to talk about two of our big projects that we work on, the Kids First project and the CBTN project, both of which touch in the rare disease world, and what types of tools we've developed to help researchers and clinicians learn more about the diseases they study. So before we get into the demo, I want to give a little bit of an introduction to who we are and what we do up here at D3B. And so we are the Center for Data-Driven Discovery in Biomedicine. And so what that means is we're a team of 65 scientists, data engineers, clinical researchers, and we are affiliated with the Division of Neurosurgery at the Children's Hospital of Philadelphia. And so there's a picture of all of us at a recent retreat back in the pre-COVID era when we could all be together in person. And so our mission is to build a transformative healthcare discovery ecosystem by providing innovative care for all children through collaborative data-driven science. And so there's a lot to unpack there, but kind of the condensed version of that is our vision is that we're looking for breakthroughs for every child, every time, everywhere. And so that's a very kind of grand statement. And so in order to be able to find breakthroughs for every child, every time, everywhere, it's a grand mission, but we realize that we can't do it alone. And so we're all very strongly about collaborative data-driven science, as we say in our mission, and that we're not trying to hog anything or not trying to keep anybody out. We're looking for collaborations. We're looking for users to get involved, and we're looking for people to help facilitate people to make discoveries and further our goal. That's what we're all about here at D3B. So when we think of D3B, what we think a lot about is this model here, this kind of pathway where we go from data, information, knowledge to impact. And so an example of this is that data, something that's data might be a sequencing result, a file with a bunch of A's, T's, C's, and G's in it. And it's a very long, file, but so there's stuff in there, but it's not very usable in its form. So that's data. It's in this kind of very original format. And so in order to be able to get the useful stuff out of there, you need to turn that data into what we call information. And so information might be a file that pulls out, specific letters out of that big text file and points out important things. And so maybe something that's information might be this participant, this patient in our research study has a mutation in this gene. So that's a piece of information. So now we're getting a little closer. We can start to learn and use that. And so if we take lots of information and we look at it together, then we start to create knowledge. And so if we get to the point where we say, okay, this patient and these 3 other participants and these 5 other participants over here, they all have the same mutation in the same gene, and we learn from our information. So now we're getting towards knowledge. So maybe this gene is involved or associated with this particular diagnosis or this particular phenotype. So that's knowledge. And then that finally leads to the impact, which is what we're trying to get there at the end. Is so now that we've learned this, now that we know that this this gene is associated with this disease or this phenotype, what can we do about it? How can we treat that? Is there any type of target we can design? How can we act on that to really have our impacts? So we think a lot about this pathway of how we move from data to information. And so it's not just a pathway, though, it's also condensed there in the middle as information, because we see that as kind of a big bottleneck of how do we get that data into information. And so we get more and more genomes every day. Sequencing is getting cheaper and cheaper. And so we get more and more data piling up, but we don't have the ability or as fast of an ability to turn that data into information. And so that's kind of our bottleneck. And so how do we ease that bottleneck? And so at the center, we work on kind of two different sides of it. People on the left side of it, a lot of our data engineers and our computer scientists, they're very interested. How do we turn that data into information more efficiently? How do we turn over more genomes, get more of these variants called? How do we do that even quicker? And so what I work on at the center is a little bit more on the other side of the bottleneck, is how do we disseminate that information and facilitate its transformation into knowledge? Basically, how do we get that information that we have and start spreading it out into the world and getting more eyes and more people looking at it in order to be able to facilitate this transition into impact? So that's me. So my name's David Higgins, and so my job title is I'm the Scientific Community Program Manager, which is a bit of a mouthful, but what that means in plain terms is that I'm a geneticist and I lead D3B's outreach and support team. And so my role is really focused on that transition from information to knowledge. And so as part of that, I build collaborations with our external partners from the center. I answer questions about our data sets, and I provide training about the platforms similar to what the demo is going to be for most of the talk today. So ultimately, what I want and what my job is that I want to help other researchers and clinicians in their development of impacts for rare childhood diseases. And so try to facilitate that, and as I said, get more eyes and get more people looking at the things that we've discovered so far, and the information that we've put together. Okay, so as part of that, we have two main collaborative projects, and so this goes back to the title of the talk. And so we operate as two main projects. We work on the Gabriella Miller Kids First Data Resource Center as one of our main projects, and the Children's Brain Tumor Network, which is our second project. So both of these are projects that we organize here at D3B that we are the main part of. So part of Kids First, so I'm going to talk briefly about Kids First. So Kids First, as the Kids First Data Resource Center, our specific mission with this project is that we want to alleviate suffering from childhood cancer and structural birth defects by fostering, again, more collaborative research in order to be able to uncover etiology of those diseases and support data sharing within the pediatric research community. So what we found, so this is listed as both childhood cancer and structural birth defects. And so what we found is that a participant that has a structural birth defect, maybe a cleft lip or cleft palate, will also be more likely than a peer who doesn't have that birth defect to develop a tumor, to develop a brain tumor. And so what that tells us is that we think that there are probably underlying similarities there between these two different diagnoses, even though at first glance they would seem very unrelated. And so as part of that, what we do is we work with clinicians around the country who have different data sets as part of their clinical trials. And so they might be collecting, if they're a cancer study, then they might have somatic tissue, like a tumor that was taken out as part of an operation. If it's a birth defect, then we'll have a DNA collected from a saliva sample. And what we do is we'll sequence the genomes of those samples in order to be able to try to find those underlying variants or underlying differences in the genomes that might be caused by those. And so we've been at this for quite a bit now. And so far we've released over 56,000 files associated with these different data sets for our structural birth defects and our different pediatric cancer studies. We've had more than 13,000 participants, and we have over a petabyte of data released on our portal. But we're not done, which is even more excited. And so that's just all that we've done so far, but we've already received funding for another 21,700 samples for even more different birth defects and even more different types of cancer. And so these are the projects that we're working on currently. So they're not currently released on the portal, but they are coming soon as far as data sets that are available for these different disease types. So that's one of our projects. The second project is a similar type of project called the CBTN, the Children's Brain Tumor Network. So the Children's Brain Tumor Network, we are also partnering with researchers, clinicians, and patients. And we work, this is an even larger collaboration. This is a global collaboration. And we're dedicated to creating a world where no child dies or suffers from a brain tumor. And so our core values is that we believe firmly in open science. And so again, building collaborations in order to do this. This is a task that's too big for us to handle, for any one group to handle. And so we need everybody we can possibly get to help out and share our data and get their eyes on what we've been working on. So open data and a global inclusion. So we have collaborators from around the world, from Europe and Asia, and across the United States and Canada as well. So it's a global inclusion. Anyone can participate, and of course our pioneering spirit. So this is a very large endeavor in order to be able to get such a huge scope of folks all working together. But we believe that we can do it by working hard and working with our groups. So again, we're very serious about collaboration. And so in that traditional research model, a single participant at a single institution, that institution might collect a very small number of samples each month, these being perhaps tumor samples. And so then they would only have one project, and then that data is locked up or siloed or unavailable until the results come out and it's published in an academic journal. And so our model is to really modernize and build on that older traditional model and say, no, we're going to work together. And so we have our CBTN member institutions, which are hospitals around the world, and we have more than 36,000 participants. And so because of that, we can collect many more specimens because we have such a larger network. We're all working together. We're all sharing our resources. So that means we can fuel more projects. So that means more projects, discovery projects are coming online that we can support. And then this data is available in real time. So we're not locking it up and waiting for a publication to come out. We're ready to share it out and get it out with the world. So we have to work together in order to be able to get all this done. And so we've been at this for a while too. So since 2011, we've supported more than 70 research projects from our biospecimen tissue repository. Those are our tumor tissue samples and cell lines. And we've supported more than 120 research projects from our genomics data sets. Those are the alignments and the reads and the variants. And those are the types of data sets that I'm going to go into in the second half of our talk. Okay, so those are our two main projects. So the Kids First DRC and the Children's Brain Tumor Network, CBTN. So we do all these things. So how do we actually, where do I go? How do we support all this research? And so we've built 3 different computing platforms or 3 different websites in order to be able to facilitate all this collaboration and discovery. And so this is where we're going to spend most of the talk today, is that I'm going to be giving a live demonstration of these 3 websites and how they can be used in order to be able to fuel and facilitate your own research and discovery into these rare diseases for children. So we're going to start with the Gabriella Miller Kids First Data Resource Portal. And so what that portal is for searching data sets. And so that might be a good place to start, where you want to see, well, what's available. And so as you've seen, we have a lot of different collaborations that we have. And so the portal is a good way to really search and see what's there in order to be able to make decisions a little bit further about what types of discoveries and where you want to go as far as our data that we have. At that point, we have 2 different places you might go after that, one of which is the PCBio portal. This is for reviewing genomic information about participants in a very accessible format. And so I'll give you a demonstration of that platform as well. And then finally, our third platform is Cavatica. And so Cavatica is a very powerful bioinformatic cloud research workshop. And so it's for doing these large scale bioinformatic analyses on either our samples or on your own samples as well. And so we'll talk about that when we get to near the end of our talk. Okay. And so today we're going to walk through a couple different websites, as I mentioned. So first, we're going to look at the Kids First portal to identify a cohort of participants that we might want to investigate further. After that, we're going to look in the PETSI Bio portal to review some genomic information about those participants together. And then we're going to wrap up by looking into Cavatica as how we might run a deeper analysis of available genomic files for that participant. And then hopefully, if there's some time, we might have some time for questions there at the end. So that's our outline for today's session. Okay, so the first thing we're going to go to is we're going to look at the Kids First portal. This allows you to be able to search different data sets and participants. And so a use case here for using the portal is that maybe I want to identify participants that have specific clinical phenotypes or diagnoses. And so if you're a researcher who is studying a particular diagnosis, if you're a clinician who has a patient about a particular diagnosis, and you want to find other genomic data sets that are related to that or associated with that diagnosis, this might be a good place to start. At that point, we're going to build a cohort of those participants. So, kind of collect those participants, and then we're going to see what's available, what data do we have for them. So we'll be able to explore some genomic data in PETSI, or we can identify some data files that we might want to analyze in Cavatica. Okay, so at this point, I'm going to exit my slides here because we're going to go on our first tour here. And so we're going to go to the portal, the Kids First portal. And that URL is portal.kidsfirstdrc.org. And so one thing before I jump too much into the portal, I want to say that all of the links for today, I've already put them into a PDF document that's been attached to this session on the 24 Hours of Rare website. And so if you're scribbling, they're already there, and so ready for you to click at. If you forget a name or forget to write anything down, you can visit those later, and you'll be able to get to these same websites that I'm showing today. Okay, so portal.kidsfirstdrc.org. So when you first log into first go to the Kids First portal, it will ask you to make an account. It is completely free to make an account. Anyone can make an account. If you have any Google ID, a Gmail address, then you can just type in your Google address and it will allow you in, and you'll be able to come into the portal. So anyone is allowed into the portal. Anyone can browse the different files that are available. And some of our files are open access. Some of our files are more controlled access. So either they require approval through NIH, through dbGaP, or for some CBTN files through our data use agreement, as far as our sharing agreement with our collaborators. But anyone can browse and see what's available. So anybody can do the actions that I'm going to show you today. So when you first log on to the portal, it takes you here to the dashboard, which gives you some information about different members who are here on the site, and different data sets. And so we're gonna explore some data. And so that sounds like a great place to start. Might be the Explore Data tool, which is this second tab right up here at the top. So let me get that set up. So Explore Data is exactly for what it says it is. It lets you explore the data. And so if we scroll down a little bit further, we can see we see some bar charts, we see some pie charts. There's some data. Okay, so what does all this mean and what's happening here? So what we're looking at here is this set of numbers. Is right now we're looking at data for the 18,000 participants that we have here in the Kids First portal, and that represents about 5,000 families and more than 89,000 files. Okay, so that's a lot. So that's too much to probably work with, for one individual researcher or one research team. So how do we narrow that down a little bit further in order to be able to find, files that we might look for? So that way we're not looking at 18,000 files or 18,000 participants all at once. And so what we can do is we can build queries and kind of narrow that 18,000 down to a much smaller number. And so one way you can do that is you can actually click. These are all clickable bar charts and pie charts, and that narrows the information down. So for example, if I'm interested in neuroblastoma, I could click this and we see hover over the bar chart here. It says that we have 772 participants that were diagnosed with neuroblastoma in our portal. I can click that button and now you see all my bar charts just changed. And if I scroll up here, we've added a term to this query. So now we're only looking for participants where the diagnosis is neuroblastoma. And you can see my number has gone down. Now I'm looking at 772 participants, about 13,000 files. So a little more manageable, but still probably too much for me to handle. So we can narrow it down even further. So this is our observed phenotype wheel. And it's color coded, and each of these different colors corresponds to a different phenotype. So different abnormalities that might be associated with different systems within the body. And so if I'm also interested in, so maybe I have, neuroblastoma, but I have a patient who also has a cardiovascular defect. And so are there, of these 772 patients, are there any that also have a cardiovascular defect associated with them? So again, I can click down here and begin to narrow in on this search, and says, yes, there are also 2 participants that have an abnormality of the cardiovascular system. So I can add this term to the query here, and you can see my numbers updated. So now I've narrowed down again. Now I'm working with 2 participants and 56 files. Okay, so we're getting a little more closer to a number that I can handle. And so we can save that. And so I'll show you some other ways to build these terms. So we can save that query right here and then start a new query below it. But there's other ways rather than using the interface below. And so you can also use our HPO browser, which is this tool right here, which lets you, again, narrow by those different filters. Here it lists them all out rather than in kind of the picture format. But depending on what type of diagnosis you're interested in learning more about, you can begin to choose. And so maybe since we're a brain tumor, we have lots of sight, we have a lot of abnormalities of the nervous system. And then you can continue to narrow that down, the nervous system morphology or the physiology. And then you can begin to narrow that down even further, and it gives you more and more specific terms. And so it's a hierarchy of these terms. So if I want if I want to cast a wide net, I can click this button up top and get all participants that include these subterms. Whereas if I wanted to be more specific, if I just want abnormality of brain morphology, I can click that minor, smaller term, and then bring it over here, and then apply that to my data set. And so now I've got this new, cohort where I've got 110 participants that have that abnormality of the brain. We also have a search box as well, and so we've got these drop downs which give lots of different information and different filters that you can apply, but we've also got a search box as well. And so if you are interested in a particular study, so like our microsomia study or craniopharyngioma, particular diagnosis, you can click apply, and now we're getting the 130 craniopharyngioma participants. Okay, so that lets you query and explore. And so I'm going to go back to my first query here, since it got us down to 2 participants. And so let's learn a little bit more about those 2 participants. And so right now we've got this kind of summary view with all of these flashy graphics, but if we switch over to the table view, it gives us a little bit more information about who those participants are. So all of our information has been de-identified, so there's nothing that's going to link any patient back to their name or where their location is, but we do have some demographics as far as the child's sex, the child's vital status, the diagnoses, and usually the age of the child at these particular events were diagnosed at as well. So our search result came up with 2, and so here are our 2 participants. And it gives us some summary information. You can even click that participant ID. So let's say I want to learn more about this second participant right here. It gives us more information about that participant. And so it tells us this is their ID number. It gives us the race and ethnicity information. And then down here has information of different biospecimens that we've received as part of CBTN. So some of these, so this particular biospecimen, this is a a tissue sample. This is tumor tissue from a tumor progression from this particular patient. And so it was a frozen tumor tissue. And so if researchers are interested in collaborating and doing a study on the tissue itself, we may be able, that you can reach out to us at CBTN through our CBTN network, and we may be able to acquire it and send that tissue as part of our collaboration. So it allows researchers to be able to explore our biospecimens and also look at our genomic data as well. So here we've got some aligned reads, so some genome sequences, and then we've got some called variants as well for this particular participant. And then further, we've got some additional data sources. We've got some images. We've got an operation report from a surgeon who operated on this child. We've got a pathology report from the histologist who was looking at the histology images. And so we've got more information provided and available. Okay, so I'm going to back up here, and I'm going to stop at this point. So at this point, we're kind of at a, I don't want to say a fork in the road because it's not a hard choice, you have to go one or the other, but now there's kind of a decision to make of, well, where do I go next? What do I do? So these are, this child, I want to learn more about this child. Maybe I'm treating a patient that's similar to this child. How can I learn more? And so at this point, this brings us to these two links down here below, one of which takes us to our other website, to PEDC Bio Portal, and the other brings up the files in the file repository, which can then lead us to Cavatica. So that takes us down a different path. So I'm actually going to pop back to my slides real quick and show one more slide. And so we've explored the Kids First portal, and we found these participants. So where do I go next? How do I make that decision? And so this is kind of the to help you decide, where you want to go. You can either go towards the PEDC Bio portal or into Cavatica. And so if you are looking for access to the pre-analyzed data, so you're not looking to do an intense analysis yourself, you want to look at the variants that are already there, then PEDC is the tool for you, because it's already available. It's in a very visual format that we're going to see here in a second. Excuse me, and it actually allows you to be able to generate graphs and compare participants as well. And so my word that I like to use for PEDC Bio Portal is either easy or accessible. It's very accessible. We want to try anybody who can go into a browser and point and click can use PEDC Bio Portal. The other tool, Cavatica, is our powerful tool. And so this is where, if you are have a bioinformatic experience, if you want to analyze our data further and one of these large, powerful, computer pipelines, maybe you have your own samples. And so maybe you want to, you found our samples through CBTN, but you have your own data set that you want to compare, but you only have a handful. And so maybe if we combine them together, then we get a larger data set and we can make more discoveries. And so if you want to run your own analysis in order to be able to kind of compare them to ours, you can do that in Cavatica as well. So Cavatica is a very powerful tool. So that's my key word that I like to use with Cavatica. It's powerful. Okay, so these are our two different places. And so as you're sitting there, you might think, I could see value in both of those, or maybe, one of these is probably more of what I'm looking for. So let me show you a little bit more in detail of what these look like, and then that can help you make the decision as well. So the first place I'm going to go is I'm going to go towards PEDC Bio Portal. And so for this particular participant, I can click this button right here, and it's going to open a separate page. And so now we're into PEDC Bio Portal. And so what PEDC Bio Portal is it's a demonstration or a visualization tool. And so what we've done is we've taken those genomic files and we've taken those variant files that we've generated, and we've taken that long list of this huge text file with the ones and zeros, and we've translated it into a plain language that hopefully people can understand. And so what we're looking at here, this is our patient with the same PT ID number and the neuroblastoma case. And so what we can look at is this is what we learned from sequencing that particular tumor. So all of our data in PEDC Bio is somatic data. So this is information from the tumor itself. And so this is, try to identify, well, where did these cells, how did they go down this path of becoming cancerous tumor cells? And so we sequenced those cells, the DNA inside those cells, and this is what we found. And so this table right here, so it gives us an overview of those different mutations. And so here's our gene. And so our top hit, it's sorted by this annotation tool, this gene ALK, and we found this protein change, F1174L, in it. And so for some people that might be, well, what does that even mean? You know, what does that even mean? Is that, yeah. And so that's why we've included this annotation tab right here to kind of spell that out for you. And so we take this mutation that we found and we say, well, what have other people found about it? You know, we're not the only ones who study cancer. What have other researchers, has anybody else seen this out here? And we reference other databases, and that's what these annotations are referring to. So if I hover over this annotation, oh, here's some more information to read. Okay, so the ALK F1174L in neuroblastoma. Okay, so what is ALK? So it tells me ALK is a receptor tyrosine kinase. It's currently altered by chromosomal rearrangements in various cancer types, including large cell lymphoma, non-small cell lung cancer, and inflammatory myofibroblastic tumors. Okay, and then this even further more to the succinct point, the ALK F1174L mutation is known to be oncogenic. And so it gives us that information immediately that, you know, not just that this child has cancer and not just that this child has neuroblastoma, but this is probably where, you know, the key mutation that led these cells down this pathway in the development of this tumor, this particular mutation. And so we've got information about that particular mutation as well. And so what that allows researchers to do at this point is that a user can say, so for example, as part of our CBTN data, a user can reach out to us and say, I see that you have a participant that has this ALK mutation, and I'm treating a patient that has an ALK mutation too. Can you help me? You know, what more do you know about that patient? How is that patient treated? What were the outcomes of that patient? And so that's information that we have. That information is not on PC Bio Portal, but we are willing to share that with our collaborators through our data use agreements. And so that would be that kind of last step is through an interaction by reaching out to us through CBTN, and we're willing to share that data in order to help direct not just research but treatment as well. So, PC Bio Portal, you don't have to get here. This doesn't have to be the second place you go. You can start at PC Bio Portal as well by going to our URL, pcbioportal.kidsfirstdrc.org. And so I'll even log out here to show you the login screen. And so if you go to this URL, it will bring you to this page, which asks you to log in. And so what I love about PC Bio Portal is that everything on the website is 100% open access. So everything that anybody is watching, if you log in with a Google account, with a free account, you can look at the same information that I'm looking at. You can click on the same buttons that I'm clicking at, and everything is right there available to you. Everything is open access that we're going to see on this website. So I'm going to log in with Google, and I'm going to use my address here, and it's going to log in. And oh no, what is happening? Let's try resubmitting that. Okay. Let's try this one more time. Oh no. Okay. So this is our CBio portal, our PC Bio portal. I think I know what's going on, but I don't have time to fix it right now. But the good thing about PC Bio Portal is that I can actually show you another website that we collaborate with. This is called CBio Portal. So CBio Portal, if you'll notice, it's actually nearly the same site. And so the stand-up is exactly the same. It's the same interface. The same tools are in here, but it actually has different data sets. And so the PC Bio Portal tool that we build is based on pediatric data as part of our pediatric research. The CBio Portal is an even larger set because it includes some pediatric cancer studies, but also adult cancers as well. And so if you're studying rare diseases but are not necessarily interested in pediatrics, then the CBio portal might be the place for you because it allows you to be able to browse these different data sets. And so I'll just show a brief demonstration of what this looks like. And so we can look at, for example, let's see here, we can look at a a neuroblastoma, since we were looking at some of those earlier. And we can, if we had a particular gene that we study, so maybe we know that we study that ALK gene that we discovered, in our previous query, we can click the query by gene and it gives us a little search box, and we can type ALK in that box and it will give us more information. Or if we're at the beginning of our search, we can just explore and click that explore button and see what happens. And so that brings us to this exploration page, where it begins to provide us lots of different information about these patients in this particular study. So this is a neuroblastoma study. We're looking at whole genome sequencing on over 1,000 neuroblastoma patients. And so it tells us information, so in our different mutated genes, and so in our different studies. This is different. CNA is copy number. So this is number where a whole gene might get duplicated or a whole gene might be deleted. So we're changing the number of copies. And so what this frequency here is telling us is that's the frequency, the number of samples that have that particular mutation. So of the 1,089 participants here, there are 5 participants that have that mutation. And likewise here for this MYCN copy number alteration, there are 18.6% of participants that have this mutation associated with them. And so if we scroll down, it turns out that ALK mutation that we discovered is actually quite rare. So of our 1,000 participants in this neuroblastoma study, only one of them has a mutation in ALK. So we can explore that a little bit further and say, well, how does that compare to our mutation or our participant that we studied in our Kids First portal? So that I could go up here to the search box and I can type ALK and query it. And it takes me to the query page where I can learn more about this gene. And so what brings up is the OncoPrint tab, which gives us some information about our different participants. And so this is what this represents is a set of tracks. And so each, similar to train tracks is what these long parallel lines. And so what each of these columns represents, this represents one participant in the study. And so all the boxes in that particular column all correspond to the same participant. So then the color of the boxes, the color of the dominoes, are these different mutations that are associated with that particular track. So here on ALK, we've got, again, this is such a rare mutation. Only 0.2% of neuroblastomas cases have it. We've got 2 different colors here. We've got this one participant with the amplification, one participant with this splicing mutation, and then all of these other participants that have no alteration in this particular gene. You can explore further in the mutations tab, and so this brings you to this page where we can see those mutations on a representation of the gene itself. And so this is a mutation right down here, downstream at spot 1389, so a little bit downstream of where we were looking in PEDC bioportal. We can click on this participant and we can bring up that same page that we were looking at in PEDC, where we can see all the mutations that are associated with that particular participant. And then we can also do a comparison study as well. And so if we want to look at, how do participants that have this ALK mutation, how do they compare to the other unaltered group or the other 1,000 participants in that study? And so we could explore. So we're a little lopsided. So I'm hesitant to look at this comparison too closely because our sample size is so dramatically different from comparing 2 to 1,000. But you can begin to look at things like survival. You can look at clinical information as well. And it does the comparison and it does the statistics for you right here in the browser. So everything is point and click and everything is accessible. So this is the CBio portal tool. So I wanted to introduce CBio portal to you anyway, because it introduces an even larger set as part of Rare Disease Day. There are rare diseases that affect more than just children. And so CBio portal is the place to go. And PEDC portal is our website, which uses the same interface, the same point and click, the same menus as we have. So I point you towards either of those great websites as a really accessible tool to be able to learn more about particular participants. Okay. So at that point, if maybe we want to go a little bit deeper, and so we've explored PTSI and we found this participant, and maybe we want to learn a little bit more about the genome files for that participant. So now this would be a great place to go for our third platform, which is Cavatica. So Cavatica is D3B's cloud analysis platform. And so what that means is that this is not just a place to query, not just a place to search, but this is the place to do your research. This is the place to do your analysis in the cloud, in your browser. And so, to get into Cavatica, you bring your data in, and that can be brought from the Kids First portal, as I'm going to show here in a second. But users can also bring their own data into Cavatica too. And so that use case I mentioned of, maybe at your hospital or maybe at your institution, you have a set of samples from a study, but maybe it's not enough to be able to do your analysis. And so if you have a couple, but we have more, we can compare notes, we can share, we can all look across all of those different numbers and help, further your research. And so you can bring your data with you and compare it to our data from the Kids First portal and bring it into Cavatica. From there, you can begin to work on what are called workflows. And so, begin to process those files, those computer files. And so users can either select from a library of existing bioinformatics pipelines that we and other users have developed on Cavatica. If you're computer savvy, you can develop your own pipeline. If you have a very specific experiment that you want to run, the tools are there to help you to be able to do that. And then finally, at the end, the end of the experiment doesn't end with the end of the workflow. You still need to maybe explore that information further, generate some graphs, dive a little bit deeper. And so that's where the data cruncher tool comes in. And so we support Jupyter notebooks for Python-based analysis, as well as RStudio for R-based analysis. And so if you already have your R script that you like to run, you can bring that up into Cavatica, double click and open RStudio, and you can run that script right in the browser on our data, on our files, without having to download all of these massive genome files onto your local computer or onto your local cluster at your institution. So Cavatica, before we show it, and I'll show it briefly here at the end. So Cavatica is organized by what are called projects. So as we said, Cavatica is a place to do work and do your research. And so, for example, I might have a project called my craniopharyngioma collaboration that I'm working on with some collaborators. And so within that project, there are members associated with that project. So those are users who are working on the project. And what that does is it allows you to be able to collaborate across institutions very easily. And so if I find somebody that I want to add, either my coworker here at CHOP or my collaborator over in Italy, I can add them to that project and they'll be able to see the same things that I do. And so it allows for that very easy collaboration, which, as you've heard, is what we're all about at D3B. It also allows you to secure your data, so you're only giving access to people that you trust, and so it's a secure environment. You don't need you're only giving the keys to people that you know and trust as part of your collaboration. So within your project, you're also going to have files, and so these are data files that are being studied as part of your project. And so, for example, you could load CBTN data from within Cavatica itself or from the Kids First portal. It also allows you to be able to bring your own data, as we mentioned. So either uploading it from within the browser via API or by loading an AWS S3 volume. So once we have those files, though, what do we do with them? And so these are the apps. So like the app store on your phone, we have an app repository. It's not a store because everything is available. Our 450 public apps are available for very common bioinformatic workflows. And as we said, if you are savvy or have your own pipeline, you can bring your own and create your own app using either Docker tools or the common workflow language. And so then finally, when you take those files and you run them through an app, you generate what's called a task, which is an instance of running an app. And so tasks are great because that information is maintained on the platform in Cavatica for future use. So any work that you do is saved right there, just like your virtual lab notebook, but it's already written down for you. You don't even have to take the notes; it's right there. And so the tasks and the files are traceable, so you can go back and see, who ran this. Was it David? Was it his coworker? Was it our collaborator? Who did it? And it's also reproducible as well. So then you can go back months later and say, oh, remember that study that we were working on, and what did we do? What was the analysis that we did? You can pull up, oh, here it is. We can pull it up here, that old task, and click a button and say, yep, this looks great. Let's run this again here today, and get that same information just copied over, and maybe plug in some new files. So it's traceable and reproducible. And then Cavatica, it supports more than just those one project though, so you can have multiple projects. So maybe I also want a space where I just want to work by myself, my own prep space, where I can have my own files and my own apps that I'm developing. And then maybe I'm also involved in a study on low-grade glioma, and so I can have a separate project with those people. But each project is its own separate workspace, and so the members and files are all kind of categorized into these different projects, and so it allows you to be able to keep things separate towards your specific particular work. Okay, I believe that's my last Cavatica slide. Yes. So now let's go into Cavatica here with maybe our last couple minutes, and then I will wrap up and take some time for questions. Okay, so before we go back to Cavatica, I'm actually going to go back to our first tool, the Kids First portal. And so here we said that we looked at this participant in PC Bio portal. So now let's say, okay, let's look at these files, these 26 files. And so let's click this and see what files are available for this participant. So what we did was I clicked that button and we've moved from Explore Data. Now we're in the file repository. So now we're looking at the files, the genome files that we have for this particular participant. And so we can see here, we can continue to narrow further. So similar on Explore Data, we can narrow by clinical filters in this tool as well. But I'm going to skip over to the file filters here and begin to kind of explore. So if we're looking for genome files, we can pull up the whole genome files. We also, for this particular participant, we have some RNA sequencing files as well. We could explore that. I can check this box and then it narrows down the information. Everything updates live as I click these particular buttons. You can also explore by different data types. And so if if I'm looking for alignments for the genomes, I can click the aligned reads and it brings up the alignments in BAM and CRAM format. It narrows that down. If I am looking for histology images, so images of this tumor under the microscope, I can click this slide and get those images. And then I can also read the report written by the pathology report written by that person at that hospital who was looking at this patient and see their thoughts, their interpretation of those images as well. So it's all here in the portal. And we have different information for different participants. So for some participants, we also have radiology images as well. So we have MRI images. And we have all different kinds of both raw data in terms of the aligned reads as well as processed data. So for example, these copy number variations for our different genome files. Okay, so let's start with, so let's say I'm looking for an alignment. So I want to start with the aligned reads. I want to maybe do my own process and start early up in the workflow here. And so here's this particular participant. It looks like we've got 7 different alignment files for this particular participant. And for the sake of example, I'm just going to choose the top 2 right here and say these are the ones that I want to study further. We can get more information about what these files are by clicking the file ID. And this will bring up more information. It will tell us that this particular sample, so this is a sequence from an RNA alignment, and this is tumor tissue, and it's aligned to the HG19 genome. So it gives us some more information about what we're getting in here. We're not just completely blind about what these files are. You can also download a file manifest, and so that generates an Excel sheet that you can look at that includes that same information about RNA, DNA, reference genome, things like that. You can download clinical data about these participants. You can download biospecimen data about this particular biospecimen. And so it doesn't, we don't like to overwhelm people on this screen, but we do have a lot of information that's available through these download buttons. Okay, so I've decided that these two are the ones that I want to work with. So let's analyze those in Cavatica. So at this point, you could download them to your computer, but hey, these are pretty big. You know, both of these files are in the teens of gigabases, so that would take a long time to try to download to my computer, and would quickly fill up if I'm looking at more than 1 or 2 patients on my computer. So this is the strength of working in the cloud, is that you don't have to download these files. You can do it all on the cloud in your Genome browser. So I'm gonna, rather than download these files to my laptop here, I'm gonna analyze them in Cavatica. And so I've checked those 2 boxes off, and I'm gonna click the Analyze in Cavatica button. And it says, which Cavatica project do you want to copy these to? And so I'm involved in a couple different projects here, but I have a demo project that I use for my demos like this. So I'm gonna copy these 2 files over to there. And it's going to say, success, the files were copied to your Cavatica project, Higgins demo. Do you want to open it in Cavatica? Yes, that sounds great. So I'll click this Open Project in Cavatica button. It's going to ask me to log in. And it's going to confirm that I'm not a bad person and I'm doing the right things. And I'm going to say yes. Okay, and so here we are. Now we are in Cavatica. And so Cavatica, as I said, it's our powerful tool. And so it's not as maybe as visually intuitive as some of our others, but this is the place where you can do the very powerful stuff. So when you open your project in Cavatica, you get this description tab. So this is, I'm just using this as a demo. So I have some of that name information that I gave in my slides. And then here are all the members that are associated with that project. And so right now it's just me, and I'm all alone here because it's just my demo. But if I want to, I could invite maybe my coworker, Young Kuhn, who is one of our bioinformaticians, and he's a really smart guy and knows a lot about bioinformatics. So he would be a great collaborator to invite here. So I could click his name and give him and invite him to this project, and then he would pop up right here next to me, and we would be able to see the same things together. We've also got, so I can click over to our files, and here are those 2 files that I sent over from the portal. So those 2 RNA-seq alignments that I pushed over from the portal are now appeared here. They're ready for me to go. They're ready for me to analyze them. So how would I analyze them? Maybe I would choose a particular app. And so our apps are in the, we have a couple here that I've already loaded. I could also add an app as well. And so this is kind of our app store, but again, this is our public app. So everything here is free to access, at least in terms of using the code. So I can show you some of our apps that we've developed. So if you search for Kids First, these are the Kids First DRC pipelines. And so these are actually the same tools that we use to generate all the files that we're looking at, all the genome files that we're looking at on the Kids First portal. So we use Cavatica ourselves, and so we're firm believers in the platform and in the power of the cloud, and using that as a way to facilitate and transfer this knowledge and information. And so our tools are available, and this is where I was saying, if you have your own samples that you want to study, your own BAM files, and you want to compare them to ours, you can bring them into Cavatica. You can run them on one of our pipelines, and then the output will be apples to apples. You can do that comparison and really kind of compare. So maybe since I chose RNA sequencing, we can take a look at the RNA sequencing workflow. And so if I click that button, it gives us some more information about it, about this particular tool. And so this pipeline, it gives us some information about the different software tools that the file is manipulated through and it runs through in order to be able to identify that information. It gives us some runtime estimates as well. And so in order to do this computing, we need to spin up some computers. And so that takes time. And so for this particular study, we could run an 8 GB single N FastQ input. It would take about an hour to run, and it would cost about $2. So all users on Cavatica are given $100 to begin their exploration of the tool. And we can get in touch as far as either collaborating and finding more funds or setting up your own billing group with Cavatica in order to be able to pay for the compute costs associated with these large scale analyses. And so then the last thing I'll point out is I'll go back to my project here, and I will go back to the dashboard here, and I'll show down here in the bottom, these are my tasks. And so at this point, my tasks, I ran this task, this BAM tools index run, and you can see I ran it all the way back on May 29th. So we're coming up on over a year here, and I definitely don't remember all the settings of what I selected for that last May. But thankfully, if we click on this, it gives us information. And so it says, yep, you ran it on May 29th. It took about 5 minutes and it only cost 3 cents. This is a very small tool. This was my input that I plugged into it, and this was the output file that it produced, and it generated an index for that particular file. So it's all right here, you know, and then if I want to say, oh yeah, I want to do the same thing again, I want to generate another index, I can click the play button, and it will allow me to be able to fill in a blank and plug in my own sequence. So maybe now I want to use my new file that I just got, my RNA-seq file that I just got from the portal, and I want to plug that in right there, and I will select no value on my parameters, and I can click that run button, and it will begin that process of building the index file for this particular alignment. So that's Cavatica in a nutshell. And so that is pretty much all I've prepared, and I have about maybe a couple minutes left here at the end. But maybe let me wrap up here by saying thank you so much for inviting me here today. I've had a great time presenting, and as I said, my whole position is trying to get more people to work with us and more people to talk to us and get our tools and data in as many capable hands as possible. So please reach out to me. If you have any questions about what you saw, if you're looking for technical support on how to use one of these sites, if you want a follow-up where you want a one-on-one session where we can talk in more detail, I'd love to schedule that with anybody who reaches out. And so our main place to get in contact with us is the support inbox, support@kidsfirsttrc.org. So lots of us at the center see that, including me. And so I'll see your email in there and I'll be able to reach out to you from that point. If you're looking for more information about us or any of our projects, you can learn more about D3B itself here at CHOP at d3b.center. And then our two main projects that I mentioned, kidsfirstdrc.org and cbtn.org. And we're also on social media at kidsfirstdrc and at CBTN Network. And all the major social media platforms. And so reach out to us there, follow us, and you'll hear about the exciting things that we've got going on, different training sessions, different data sets that we're releasing, and get all the latest news as far as our different projects go. And so with that, I think I'm gonna stop talking here and say thank you so much again for having me. And if there's a little bit of time, I'll be happy to take some questions.
David, thank you. Eden apologizes. She kicked us off with you. She had to step out for just a few minutes, so I am sitting in for her. Great presentation from what I saw. Just a couple of quick questions here about really one of the main ones with what has happened with COVID over the last year. What type of impact did that have on what you're working on with Kids First?
For sure, yeah.
Help or did it hinder?
For sure, yeah. So things have definitely changed. And so on my slide there, I've got our beautiful building in Center City, Philadelphia, and there we are in the lobby in the picture. But you can see now I'm coming to you live from not that building, from my own apartment. And so a lot of the computer folks, interestingly, we've been able to keep going. We've been able to keep moving forward. And so that's really the benefit of this, this cloud environment where anybody can do it with the good internet connection. You can access these. And in fact, when everything first shut down, we actually got an increase in traffic a little bit. I think as researchers were maybe they couldn't be in the lab, and so they decided to check us out and be like, oh, what is Kids First? And I can do that, from the work from home, environment. So we did that. So our data operations side of it has been has kept moving forward. Our lab side, so we do have people who work in the lab as well, who handle the tissue specimens and the tumor, and so they have been on they've had a much harder time of it because obviously they can't work from home to do that type of work, but they've continued to keep it up, and they've been continued to receive samples. They work, strange hours depending on when the surgeon is available and when, obviously, if a child comes in, but they're there every single time ready. If a child is willing to participate and become a participant in CBTN, then we're there to receive their gift and hopefully, maximize the impact of that tissue donation as much as we can. So it has affected us, but we've been able to keep moving forward.
Well, fantastic. Thank you for sharing. I know we're out of time on this one. David, fantastic presentation. It's such an honor to have you with us. Just want you to know from the bottom of our hearts, from Eden's and mine, thank you. And thank you for the work that you're doing in this to help people.
Well, thank you so much for having me again. And as I mentioned, our mission is to collaborate. And so that's what I love about this event, is that we're getting so many people together from all different backgrounds and all different places and all different experiences. And so we want to help everybody, and we want to help kids. And so please reach out if you think we can help you, because we'll do our best.
All right.
Thanks so much for having me.
Thank you, David. Talk to you soon.
Of course. Have a good one.